guided all hydrogen energy.
Welcome, everybody. The Biotronics Laboratory at the Material Science and Engineering School at Tecnología de Costa Rica is pleased to welcome you today to this presentation by our distinguished professor, Barry Bruce, from the University of Tennessee Knoxville. The Biotronics Laboratory seeks and envisions to develop electronic architectures that are based on renewable carbon resources to reduce the environmental impact of the electronic devices that we have today, from the materials extraction and all the way through their life cycle until they become a waste. So today we are holding a hybrid event. We have some of our audience is connected through Zoom, and we also have audience on the auditorium. We have a very diverse audience. We have professors and students from electronics, biology, chemistry, and diverse areas of engineering. We also have people from the government and from industry. The dynamics, you can hear the people that it's gonna be in Zoom. If you have questions, we will answer them all at the end. The people can interact with the speaker and with the audience in here. You can ask to make a question by raising your hand, and our technician, he will allow you to speak. We also encourage the speakers and the audience that have questions to turn on their camera, and then their camera we can see here in the screen, so that we can have a more interpersonal communication and scientific discussion. And also the people here in the audience, you can interact with the people on Zoom. We have another camera here, so they can see you and they can hear you through this microphone. So now to introduce Professor Barry Bruce. Dr. Barry Bruce is the Charles P. Postel Distinguished Professor of Biochemistry. He is a professor in multiple departments, including biochemistry and cellular molecular biology, microbiology, chemical biomolecular engineering, genome science and technology, and inaugural member of the Bredesen Center Interdisciplinary Research. He has a Bachelor in Science and Biology from the University of California at Santa Cruz, and holds a Master's in Biochemistry Biophysics from the University of Massachusetts at Amherst. In 1990, he received his PhD in molecular plant biology from the University of California at Berkeley. Dr. Bruce joined the UT Knoxville faculty in 1994 after completing a National Science Foundation postdoctoral fellowship in plant biology at the University of Wisconsin at Madison. He is a fellow of the American Association for Advancement of Science. For the past 4 years, he has also been a regular speaker and instructor at Congreso Nacional de Ciencia, Tecnología y Sociedad, taking place in different parts of Costa Rica, and helping to introduce photosynthesis and sustainable energy to the science educators throughout Costa Rica. And now we have Professor Barry Bruce with you. Thank you.
Okay, thank you, Claudio. Let's see. Okay, is that good? Everybody can see this? Okay. I will use my sustainable laser pointer. Oh, no. I'm not. So this work is a field which I like to say I kind of invented this term, applied photosynthesis. And the term is to contrast with an area of research that's often called artificial photosynthesis or biomimetic photosynthesis. And our approach is to actually take the parts of the plant or the algae that are driving the natural process of photosynthesis and removing it from the plant and putting it into a new solid state device that's going to combine material science and biology. And this is the complex I work with. This is called Photosystem 1. Some of you may not know about Photosystem 1. I'll tell you a little bit about it to introduce this material. So one of the challenges that we know is that the Earth and humans and lifestyle, and especially modern lifestyle, has made us very addicted to electrical devices. I couldn't even tell you how many are in this room, but I would guess there's 25. It's estimated that approximately 10 electronic devices in the communication form, whether it's a cell phone, smartphone, or laptop, exist on the planet Earth. This amount of electricity to drive these things is going forward all the time. Surprisingly, about 25% of the planet still doesn't have access to steady electrical energies. But if you look at this, you can see that all of these industrialized nations, this is a combined solar image of nighttime lighting. And there's a lot of places in Australia, Sub-Saharan Africa, South America, that people are not yet using electricity, but this trend is expected to not stay flat, and it's going to go up. And we're going to probably increase our electrical needs by, we think, especially in the non-OECD, the non-developed nations, by almost 100% in the next, in this case, 30 years. So you can see all of these countries don't participate. Costa Rica, where we are today, is one of the members of this group. We also know that from climate change perspectives, we are trying to lower our dependence on fossil fuels, particularly coal. And it's been estimated that this green line of renewables will have to fill in this electrical energy. Now, most people, when they think of renewables, have been thinking of things like biofuels, hydroelectric, wind, things like that. The problem with those there's nice technologies. They just are not distributable worldwide. And so almost everybody has predicted that to fill in this energy need, this little yellow-green box, we're going to have to use photovoltaics. And photovoltaics is a general term for a multitude of technologies that can take sunlight, directly convert it into an electron. That electron can be used as electricity. And you can build these solar plants. You can build these things as large as you want. But one of the challenges that people have discovered is that although it will take a lot of land, and it depends on your perspective, whether photovoltaics takes a lot of land. This is a current projection. This is Spain, and this little blue box represents the amount of land that the planet Earth would have to commit, societies on Earth would have to commit to provide all of the electrical energy by photovoltaics using relatively low efficiencies, approximately 10%. Now, the goal would be not to centralize it, but would be to distribute it. And the thought is that this would not be such a burden. And one of the things that I like to reference is that if we were to just go and look at all of the golf courses where you go and go putting with golf, if you were to convert those golf courses to solar cells, solar farms, you would come pretty close to covering about 10% of the needs. If you take the highway system in the United States, about 75 years ago, the United States used eminent domain to take over a large amount of land. They made the interstate highway system, the Eisenhower Act. If you were to take that same amount of land, you would also be able to cover about 20%. So the land itself, although maybe geopolitically or politically questionable, it is not unobtainable. What is a problem, however, are the resources that go into solar cell production. Almost all of the first, second, third, and fourth generation solar cells are built around metals, rare earths, metals, and resources that simply do not have enough natural abundance. And if you look at the USGS, they have gone out and made really pretty careful predictions on what are the level of natural resources on the planet Earth that are obtainable by current mining and strip mining techniques. And in almost every case, the projected availability of the metal falls extremely short of our energy needs projected out for 2100. And so this means that at least with the current solar cell technologies, we simply cannot just go back and build. Sorry. Okay. We just can't build these type of distributed solar cells because the raw materials are simply not available. And so what we would like to do is basically generate strategies that allow this to come up, hopefully to about a 25% photovoltaic yield by using resources that are not resource limited. Now, one of the ideas that has come forward to help satisfy this is to go and start mining deep sea mineral rich deposits. And so this is a global illustration where a lot of these polymetallic nodules shown here and these cobalt rich crusts exist. And so there's already a lot of companies that are designed to go down and do this deep sea mining. But the deep sea mining has such a tremendously negative impact for biodiversity, as well as other issues such as oceanographic currents, that there's now a ban that's been signed essentially by many companies now, BMW, Volvo, Google, Samsung. So we've essentially had a moratorium on using any of these natural resources that are in the deep sea beds. And this only extends the problem to how are we going to make this. So this is a cartoon I made several years ago to really try to push the concept that maybe we don't need to completely build a new energy converting system or another solar energy converting system. And everyone has latched onto the concept of green energy. They want to have green chemistry. They want to have all this stuff green. But what a lot of people, especially those who are not biologists and not photosynthetic scientists like myself, don't really understand that the illustration of using a leaf is not just pretty and comforting. If we think deeply about it, maybe we can use the leaf in a much more direct way and possibly plug into this life process and see if we can get electricity directly from plants. And so we all probably would say this is crazy, but if you think about it more deeply, we can probably make better use of the plants than what we are using with biofuels. So biologists have been very motivated to take an approach with using what we call biomass, and this biomass can be grown and can be converted. But if you look at the energy efficiency, the external energy conversion efficiency, this ECE or whatever, all of these processes are remarkably inefficient. None of them have even approached 1%. There have been experimental laboratory settings where they've grown microalgae, things like Chlamydomonas green algae in a really highly sort of pampered environment to where they can get approximately 5%, but they're all lower than the typical silicon. And the reason that this is that photosynthesis is a very complex set of processes. It's not just the photochemistry that a solar cell needs, but it has to go through all of these different conversion steps that essentially involve biochemistry, carbon metabolism, growth, reproduction, warranting, fighting pests and infections. But if we go back to the very early steps, the steps where the photon hits the leaf, the moment that the green is the pigment, the chlorophyll pigment, that process is remarkably efficient. And it's been estimated and re-estimated and remeasured that internal step, the internal quantum efficiency of photosynthesis is 1%, that is 100%, it approaches unity. So every photon that is absorbed can actually be used to generate an electron. That electron then goes through a complex set of electron transport steps, things that generate ATP and pyridine nucleotides. And at that point, the efficiency starts to drop. Now, one of our ideas is let's go back. Let's don't grow a plant. Let's not let the plant metamorphose into coal or fossil fuels. Let's go to this point right here. Let's see if we can take the components in the plant, out of the plant, put them into a device, and preserve this 100% quantum efficiency. So this is a pretty bold idea. The idea is that we're going to have to take biology, extract it, much like we would maybe extract a heart, wire it into a solar cell with specialized cathodes and anodes, and preserve all of that process. Now, that's going to be challenging, and we're certainly not there, but one of the challenging is, unlike biomass, you grow a field, you cut it down, you ultimately burn it, whether it's metabolically consumed by microorganisms or put it into your stove and burn it as a fuel. You grow it once and consume it. We can essentially grow this plant and we can use it for hundreds of days And we have done that. We've used one prep for almost 300 days. We may be able to do it for thousands of days. And if you look at it from a photocycle, you can go tens of millions or tens of billions of life cycles. So what we know about the process, I'll just go over this very quickly, is that as the sun comes in, we lose a little bit of stuff through transmission, reflection. Plants are not black, they are green. They're not green, well, they look green to us. They absorb only in the red and the blue. They let a lot of light through. Some of it is reflected. Some of the photochemical efficiency is less than perfect. And then you go down and you lose all of this. So by the time you get to biomass, depending on the plant, you're well under 10% efficiency. Now, the other challenge, which I like to bring up, which is something appreciated, especially in Costa Rica, is that any strategy to have a solar to fuel, in other words, if we're going to take sunlight, use some sort of biomaterial and convert it to fuel, we have to basically not contribute to any of these 9 areas that have already been exceeded for a sustainable planet. This is what we think is kind of a safe operating space here in green, but this represents biodiversity, climate change, acidification, phosphorus cycle. All of these areas, agriculture and human activities impact negatively. And virtually nobody in the biomass community can provide a scenario where we can grow enough plants on the earth to not start pushing one or more of these outside of this safe green operating zone. Now, what we would like, and so this kind of gives you the problem in a relatively graphic way, this is how much natural energy we have photosynthetically on the planet, 78 terawatts. This is essentially the bioavailable net primary productivity of all life on the planet. This is what the humans are using. Black represents all fossil fuels. This represents a little renewable, a little nuclear. What we would like to do is to stop using this black energy and basically make it a green energy. Now, if you go through the biomass approach, you can add that, but what happens is essentially the bioavailable productivity that's available for the planet gets consumed. So we're now shifting the net primary productivity from the Earth's use, whether this is carbon balance, climate change, whatever, and we're extending it to humans. And so anytime that we raise this area at the expense of this area, this is going to be probably cataclysmic events on the Earth. So what we would like is to have another system. We would like to have an artificial system or an applied system where now we add this here, but we don't move this back, okay? So now we're adding the productivity from this artificial or applied system to the primary productivity without any deleterious effects. So this is the goal. We want to basically plug directly into photosynthesis. We want to take the photosynthetic process, the living process, put it into a system where we can now make energy. So this is a cartoon that kind of summarizes the challenge. We're going to have to take the biological process that we know very well. We don't know it perfectly. We're going to have to go down this green pathway, and then we're going to have to jump over here and generate specialized metallic conductors, electrical circuits, things like that, eventually storage devices. And now by going in this direction, we can essentially combine the power, the capabilities of biological charge separation with solid state materials, new advances in nanoscience and things like that. So we really feel like there's a unique timing because biology is having a revolution in the area of synthetic biology, material science is having a revolution in nanoscience, and the combination can do something that was never available before. So as I said, this is the complex I work on, photosystem 1. It's one of the largest membrane proteins in nature. It's found in all green organisms, all photosynthetic organisms. I work mostly with cyanobacteria. These are prokaryotes, very common. I work with a thermophilic cyanobacteria that can live up to 60 degrees Celsius. You can also work with plants. We also work with spinach. One of the first solar cells we made came from photosystem 1 from spinach. And the people that got the most excited were the people who were marketing the logo for Popeye. And so Popeye you might be too young for Popeye, but Popeye put forward the idea that eating spinach can give him power. And we made a solar cell from spinach photosystem 1, and we are actually making power directly. So what we would like to do is to essentially make a solar cell that where we replace this sort of material here, this whole transport process where we're going to get charge separation and migration. We're going to take the complex out of the membrane. Now, photosystem 1 lives in a biological membrane. Photosynthesis usually sort of divides the role of the proteins. One is a light harvesting antenna. It doesn't have the power to do charge separation, but then we have a charge separator, and that can actually generate a very strong negative electron. It can be an electron donor. Turns out that a good solar cell is going to have to be sort of balanced for the solar spectrum. Nature evolved on planet Earth, so it's already evolved to be compatible with our solar spectrum. It has to be able to take the photons, generate a charge transfer state. Photosynthesis does that very well, but it also has to be wired into a device, and the device is going to have to be able to conduct the electrons or the holes, or in a real device, both of these out with some quantum efficiency. So this is really, number 3 is the challenge that the material scientists and the biologists have to accommodate. Now, this is a little bit of a complicated diagram for many of you, I'll tell you, but I will try to just illustrate. P700 is the pigment. It has an absorption at 700 nanometers. When it absorbs a single photon, it gets excited to an excited state. The free energy difference is well over 1 volt. And then if you look at each of these forward arrows, these represent a pre-structured array of electron transport acceptors that are in a rigid scaffolding of the membrane protein. And you can see that the rates in the forward direction are quite fast. 10 picoseconds, 30 picoseconds, 300 nanoseconds. The back reactions, however, the charge recombination, the problem with most solar cell designs, is much slower. It's 1,000 to 1,000,000 times slower. So what the photosynthetic device has built is it's built a very rapid, kinetically driven charge separation that ultimately ends up with these electrons far out on one edge. The beauty is that these back reactions, if you get way out here, not only have you gone across the membrane in the bilayer, but the charge recombinations are very slow. And if you get all the way to here, this little brown box, which I will talk about, I hope, called ferredoxin, virtually there's no charge recombination. So the other advantage of photosystem 1, which I show schematically right here, this would be the plane of the membrane, is that it has a large number of chlorophyll, about 100 to 120 chlorophyll per reaction center, special pair. This is the special pair that undergoes the charge transfer. These represent electron transport steps. And when the electron gets all the way to the last step, you can see that it's actually no longer in this deeply insulating membrane. The membrane is designed to be an insulating environment, but it actually sits out on the outside edge. So it becomes highly accessible to surface chemistry and surface compounds. And so what we do is we want to build this system in and use some sort of electron donor to re-reduce the hole and another set of electron transport carriers to carry it to some sort of electrode. So in this case, we may work with gold, we may work with platinum, titanium oxide, zinc oxide, we work with all of those. And we're going to engineer these components on the two sides that are going to fill in and provide the solution to what is normally the rate limiting step in photosynthesis. This will take place very rapidly on the order of nanoseconds. These steps coming in here or going out here are very slow. It's a relatively promiscuous complex. We can take electrons directly from gold. We can go through an osmium complex. We can go through an organic complex. Nature uses the protein, and this is an iron-rich protein called cytochrome C, and its acceptor is also an iron-rich and iron-sulfur-containing protein, ferredoxin. So nature uses these guys, but maybe in the laboratory we're going to use something different, something that the chemist can create. This is what it looks like. It's easily made. This is a small bioreactor. This is a 2-liter bioreactor. My lab now has 225-liter bioreactors, so we can make this stuff in nearly an industrial scale. If you go and switch to spinach, you can grow acres of the material. We isolate it by a very simple procedure using a surfactant, a detergent, not that unlike what's in your shampoo. And when you isolate this guy out, this is the trimeric complex. If you take a transmission electron microscope, you can see these very small particles very easily. If you were to go and look and solve this by cryo-electron microscopy, you would actually see that these are the individual complexes that have been detergent isolated into a little detergent micelle. So what we really want to do is we want to make sure that this reaction center works well and that we can illuminate. It turns out that P700, based on our solar spectrum, can have a theoretical upper limit of its quantum efficiency of just around 35%. So it's really quite well. Now, the first device that was made that showed this as being a practical concept, I started doing this in about year 2000. And by 2004, we published this landmark paper, and this paper's been cited, I think now, somewhere around 700 times. And we used a reaction center that we had put in engineered in a histidine tag. This is a common trick that biochemists use. We functionalized gold with this modified NTA. This is a complex that will bind to the histidine. And that allowed us to basically layer gold down on indium tin oxide, chemically functionalize it, add the reaction center. And in this device, we overlaid it with two organic complexes that acted as electron transport layers, and they helped carry the electron out from that vectorially oriented reaction center in such a way that it was allowing it to be very conductive and would give relatively low levels of current. If you look here in the dark, the device has relatively good resistance, even with a +1 or -1 volt bias. But if you put no bias across it, you can see that we get a little itty bitty amount of current. The amount of current we got then was only 400 nanoamps, not particularly high, but this was the very first proof that a biological complex can be made as a solid state photovoltaic device. So now we're trying to improve this complex, and we want to essentially increase the capacity. The strategy that we're going to have is to move into nanostructured metal oxides, zinc oxide, titanium dioxide, and we're going to try to take this complex and engineer it by molecular biology such that it will interact specifically in an oriented fashion. And so in this case, we're going to use ferredoxin. Ferredoxin naturally binds. We're going to put a synthetic peptide that's going to be specific to binding to metal oxides. And we're going to come in either with cytochrome C or some other donor to re-reduce that complex. And the system's now going to essentially be a green solid state device. Now, one of the reasons we want to go to the nanostructured metal oxides is that the gold devices and the ITO devices, and many of the devices that people have made, are so thin. They're essentially one biomolecule thick. And the light that comes in simply is not harvested, okay? You essentially have too much transmission. You can see, this is the reason we see it as green, is that we only absorb down here around 400 and above 650. There's a lot of the spectrum that is lost. And so possibly nature can give us new pigments that would fill this in. So the device design that we put forward about probably 9 years ago now was to use molecular biology to take the complex. This is what it would do in nature. Here's our membrane, here's cytochrome C, here's ferredoxin. All of these components are wired directly into the membrane protein complex. But now we're going to essentially take one of these subunits, shown here in red, and we're going to add to it a specific sequence. Here's the sequence that's shown. This is a sequence that was actually discovered through a phage display approach that molecular biologists have used. And we can come in and we can use biology and biochemistry to replace one of these subunits so that now it will bind to titanium oxide, or we can change the sequence and it can bind to zinc oxide. So we have both of these coupling chemistries. We then want to use an effective donor. So we started with these cobalt donors, and my collaborators have now been able to improve these cobalt complexes such that they're much more water soluble and much more effective. And so when we first did this, we actually went to Zurich, we went to the EPFL, and we did these experiments in Michael Gratzel's lab. And we made, at this case, I think the highest biohybrid solar cell properties. We had 362 microamps, open circuit about 0.5. and really, really, really good fill functions of about 71%. And it was a relatively robust device that would work from very low light all the way up to 1 sun. So it looks something like this. Here's our zinc oxide nanostructures. We have modified the proteins. The proteins we think stick on the outside. The idea is that we get good coating. We probably don't get as good a coating as we think. We're probably still only working at the very tips of this. We have to work on better wetting techniques and strategies to get the protein complex in deeper into the nanostructured material. Okay, so the other strategy we had that was to try to build up a thicker system is to use conductive polymers. And so here we've designed a conductive polyvinyl imidazole that has this kind of, looks almost like a heme group, but it's an osmium complex. And in that case, we essentially make a relatively amorphous gel. The gel is drop cast onto a gold-coated ITO. And in that case, the system works quite well. We're probably, compared to our monolayer, 10 to 50 PS1s thick. And it gives us quite good properties. You can see here's an IPCE curve of getting to about 0.15%. You can see that it completely superimposes the chlorophyll absorption spectrum. And if you do all these sequence of additions, you can see that it's totally dependent upon both the osmium, the PS1, and the PVI. And it shows pretty good conversions, although it's interesting, we seem to have 2 different slopes of this, which I think is telling us something about some of the kinetics of the device. And this is actually a cross-section. This is sort of an artistic view of this. Here's our PS1 sort of just living in this transparent polymer. And here you can see the silicon, the gold, and the PVI that you can't resolve the PS1 that's distributed in that. Now, I'm going to tell you a little bit about some projects, and then I will probably be almost out of time. So we don't really like using osmium. Osmium is pretty expensive. It's about half the price of gold. We'd rather go back. I'm a biologist. I would like to go back and use the natural donor, which grows easily in a test tube and can be made very cheaply. We would like to use cytochrome C, which is contains this heme group, which is an iron. And it actually is the donor that will donate to this P700. Now, what we know is that that donation step is quite slow. So we're trying to, in my lab, increase the affinity. So we're taking this protein here, PSAF, and we're taking this protein, we're doing molecular biology to make them high affinity. We've also decided to make these things polymeric, and the idea is that maybe we can tether these and we can make something that would be a naturally occurring proteinaceous nanowire that would ferry electrons. And if we were clever enough, we could wire this complex. We've made this thing up to 6 molecules. But if we were clever, we could attach it at one end here, and then we could functionalize it to the other end where it would actually interact directly with the ITO or the gold surface. And the electrons would just basically hop between these things with very high efficiency. So we're doing that. We're also going out and we're taking electron transport complexes from other organisms. And then finally, we're synthesizing this ensemble of relatively cheap cobalt complexes that can act to donate electrons. So 3 students in my lab are working very closely on each of these, either engineering the native protein using non-native proteins or using synthetic proteins to transfer. So the other way I want to talk, so I didn't spend very much, is trying to use this ferredoxin. What is the time, Claudia? So I'm done? 20 more minutes, okay. So as I said, my lab is a biology lab, and so we sort of have people working on both sides. These 2 sides, the stromal side and the luminal side, represent the electron acceptor side or the electron donor side. They're separated on 2 different sides of the membrane. Both of them are rate limiting. I sort of quickly ran over this luminal side because I usually can't talk about both of these, but I wanted to introduce a little bit of that. But I want to talk about what I'm doing now because this is the project that I'm actually trying to collaborate with people at Tech to try to use some of this molecular biology to generate device designs, which are going to have both the natural components, but also an unnatural affinity for materials that would not normally be found in a plant. So we're going to try to marry the material affinity that we can use in the laboratory to make these things as close to a solid state device as possible. And so essentially, I'm going to talk about this one little project here. What we've been doing is we've been making this in vivo attachment of PS1 by zinc oxide and titanium dioxide, the work that I did with the Gratzel lab. It works. It's really challenging because we have to do this complex reconstitution biochemistry. But if we can use ferredoxin, this is a soluble protein, and we can generate a secondary attachment design that's going to be much more labile. So the hope, you've already seen this cartoon, is that we can now, ferredoxin is a soluble protein. It binds, it comes into PS1. This is the membrane domain where the chlorophyll is. This is that little extra membranous domain that sticks out. We actually call it the stromal hump. It's a little bit like a hat on top of the membrane. And it has these iron sulfur centers, and it binds ferredoxin. Now, in nature, ferredoxin docks very transiently. It docks long enough to pick up one electron, and then it gets up and it goes away and does the redox chemistry that its role is. It's very complicated. It makes hydrogen, it makes pyridine nucleotides, it does all sorts of reduction chemistry in biology. So its affinity for PS1 is by design very weak because it has to come and go, come and go. If it was to bind very tightly, it would get reduced, but it would never go out and do the metabolism. So we're engineering this interface, and at the same time, we are putting onto it synthetic design, synthetic peptides, and these synthetic peptides can interact with some sort of material, in this case, titanium dioxide, and that titanium dioxide will allow this complex to be tight association with this and have a highly oriented fashion. And then all we have to do is get the electron in from some other direction, some other source. So as I said, we want to, TiO2 comes in two geometries, rutile and anatase. So we have to essentially find a peptide that will interact specifically probably with the anatase form. And so to do this, we use this technique that's called phage display. And this is a technique where you can screen literally tens of millions of random peptides to see which ones will bind specifically to any material. So you're essentially doing a little, it's called molecular panning. And at the end of the day, we essentially identified two peptides that are binding this, STB1 and STB2. They have some similarities you can see, but these were randomly designed. STB1 was found 22 times, the other one was only found one time. And so we're going to take that peptide, and the idea is that we call this TOBIP, titanium oxide binding peptide, and we're going to try to put that peptide onto this protein ferredoxin in such a way that now it will attach it to this complex, and it will attach it in a completely symmetric way. It won't be random, it will be very tight. And this gives you sort of some idea. This is actually a cartoon directly from Gratzel. But the idea is that we're now going to have the dye. The dye is not going to be a ruthenium dye like Gratzel used. It's going to be the PS1 itself, and it's going to transfer the electron directly into the photoanode. So the challenge we had, if you're a protein chemist, you know that proteins have two ends. They have an N terminus and a C terminus. And we don't really know where to put the titanium dioxide binding peptide, okay? We don't have a crystal structure of this complex yet. It's been very intractable to solve the crystal structure, again, because of this highly transient interaction. And so we had to use some computational tools. And so here's kind of the state of the art of what was published. Here's these iron sulfur centers that we have here, and here's our ferredoxin. So we don't really know if there's the N terminus or the C terminus, which one of these would be the best site to attach it to. So we decided to do some computational approaches. We took this is some previously published literature where a lot of mutagenesis had been done to sort of identify where the interaction surface is on this. This is called the stromal hump right here. So we had some pretty good ideas. Here's these iron sulfur centers that I talked about. So we developed an algorithm that used a tool that's called the frustratometer. And the frustratometer predicts regions of ferredoxin that are chemically unstable. And they're chemically unstable probably because their natural role is to interact with a surface of some other protein. And so we started doing molecular docking using these candidates. And you can see that here's our ferredoxin sitting on this surface. These are these electrostatically charged surfaces. So the proteins have probably some ionic interactions. We had another site here, second site here. And so we started to do computational measurements of what is the distance of these different docking complexes. And so this is the more negative this is, the more favorable the interaction is. And we also wanted to pay attention to the distances in angstroms because we wanted to keep these electron transport elements as close together to favor the speed and efficiency of electron transfer. And so we did a lot of docking, but in the end, we essentially came up with a couple different configurations. And fortunately for us, both configurations put the N and the C terminus in relatively exposed orientations. We decided that the N terminus being further on this surface up here would be the better position to attach those peptide binding domains. So this is a kind of a cartoon of what we think is going to be happening. We're going to have our protein docking, and coming out this N terminus on the far side, we're going to bind that peptide, that titanium dioxide binding peptide, such that now the titanium oxide material will be on that side. These two electron transport components are going to be close together, and this is going to favor the diffusion-free solid-state device. And so this is the construct. We put our ferredoxin, we put in a flexible linker, and we put in these different peptides that we would hope would drive the affinity to bind to this complex. And here's essentially our test where if we look at we put in our titanium dioxide nanomaterial and we spin it down the pellet. If it binds, if it doesn't bind, there'll be nothing in the pellet. It will all be in the supernatant. By quantitating these two ratios, we can determine how high the affinity is. And it turns out that LSTB1 ferredoxin has about a 50% affinity for this titanium dioxide material. And so we look like we designed a good peptide and a good design. And so we then wanted to start modifying our titanium dioxide. So I started working with a material scientist at the University of Memphis, and he started to build more variations in the titanium dioxide material. And we were able to show that one of the complexes that he made has very high efficiency. Things that we were buying commercially available were much, much less. US Research Nanomaterials rutile is very bad. The Aldridge rutile is quite low, the anatase. But the material he generated, we can get about 50%. So we have a pretty good sense of what that device will do. Now, the last thing we were trying to do is trying to engineer the titanium dioxide to be a little bit more compatible, because if you make one of these solar cells, the first thing you'll see is that only protein chlorophyll attachment is on one side. We doctor blade this to where it's about 50 microns, but we're probably only attaching the protein to the upper maybe 5 to 10 microns of it. So I started working with a physicist in England, and he has been able to make a system where he can actually generate controllable pore sizes. And you can see they can go from 20 nanometers all the way up to probably about 250 nanometers. And to give you some idea that the size of this is about 200 nanometers, and so 200 angstroms, so about 20 nanometers. So somewhere in this size from here over, we actually think we can put the protein not on the device, but in the device. So if you look at this in the cross section, it looks almost like a sort of a honeycomb-like material or a Swiss cheese-like material. And the idea is that we're going to run, instead of having a nanostructured pillars, we're going to actually electroporate the protein down into these cavities. And by doing that, we'll be able to completely increase the surface coverage of the complex. Okay, so the last thing we're going to do is we're going to try to engineer the affinity to this material. The affinity for ferredoxin for PS1 is relatively low. So we're going to use, again, molecular genetics to come in and make a series of point mutations. And these are the ones that have been highlighted here that give us the most gain in binding energy. And we started to do those and test these guys. And sure enough, we're able to start generating species that bind in solution much more tightly to photosystem 1, and they can actually be chemically cross-linked to photosystem 1 to these stromal hump subunits, PSAA and PSAD. Okay, just one last thing, and then I would like to take questions. We're mostly using naturally occurring electron transport proteins, but the field of protein engineering has evolved so much that we don't have to rely only on what nature gives us. We can actually start to make synthetic proteins that are completely outside of the natural repertoire. And so my lab has started to work with a very innovative scientist at University of Washington, Dr. David Baker, and his postdoc in India, Roy. And so this is what the natural ferredoxin looks like, but they can make a completely synthetic, novel ferredoxin that looks like this. And by combining this idea, we think we can make a ferredoxin that has the electrochemical properties of having the right midpoint potentials, but by functionalizing this synthetic gene product, we can add whatever properties we want, whether it's protein attachment sites or electrode attachment sites. And this work is ongoing. The problem we've had is we've not been able to make this complex have the low redox potential that ferredoxin has, which is about minus 450 millivolts. Everything we make is right around 0. And so we essentially are not capturing any of the redox energy that PS1 does because there's too large of an energy gap between the donor and this synthetic acceptor. But we're continuing on with that. So I'm not completely done, but we are hopeful. So I just want to give you a little cartoon of what's happening. This is a somewhat outdated slide, but this is a graph of what has been reported to date on biohybrid solar cells, where we're looking at reported current. We normalize all this to microamps per centimeter. You can see that if you, a couple outliers here, but if you get the rest of these, you get a pretty nice linear slope. It's actually, our progress is exceeding Moore's law, the number of capacitors that you can put into a computer chip. And it has really been generated by only maybe a dozen labs. I'm very happy that we're going to add one to that. Dr. Villarreal is going to help us make better cathodes and electrodes. And then you can see a lot of these accomplishments, all the ones in yellow are work that has come from my lab. And one final slide is that we actually did this measurement. We think it's a promising capability. If you grow 1 acre of spinach, you can actually make 55 acres of solar cells. And so you're not going to have to allocate the land that we use for biomass. And this is actually a picture I took. I started a small company in the Salinas Valley called Fresh Power. And Fresh Power's idea was to repurpose spinach and make solar cells built around the natural photosynthetic process. And I just want to thank my hosts, not just for today. As I said, I've been coming to Costa Rica for the last 8 years, often with Ciencia. I have been sponsored by the National University of Costa Rica, this trip to Tech. I want to thank all of these hosts that I have had. Ale has been the director of Ciencia for forever. She's been my host. Natalie's been my host. And of course, our local host, Claudia, has been very gracious and was able to spend a few weeks in my lab before I came here. And I want to also thank the support from Copa Ande. Okay, thank you.
Well, thank you, Professor Bruce. I really had a great time in his laboratory as well. I was learning about how they build all these devices with Photosystem 1. We work in our Biotronics laboratory with the bacteriorhodopsin, that is another protein. But my protein only has one reaction center. He said that his has like 100. But I hope that our collaboration is it produces a lot of positive results. So we would like to open now the microphone for questions from the audience in general. I don't know if you can raise your hand and make questions. You can turn on our technical host. He will allow you to open your microphone and make questions, or if there are questions from the audience here today. Yes. So I'm gonna walk there and okay, go ahead. So your name and what is your department?
Yeah, my name is Fabian Cordero. I am studying materials engineering, and I would like to ask if all the plants has the same efficiency. We're talking about photosynthesis. Is the same for all, or it could change?
Okay, so efficiency in photosynthesis is always a very complicated question. So we have two types of plants that do photosynthesis. One is called a C3 plant and one is called a C4 plant. I sort of showed you on that cartoon. And they fix carbon in slightly different ways. One fixes a 3-carbon compound, another one fixes a 4-carbon compound. Those two plants, for reasons that are a little outside the scope of this talk, vary in their efficiency, okay? But that's at the final level. That's the complete growth efficiency of the plant. If you go back to the photosynthetic steps, the early steps, virtually these processes are identical in every I'm going to add a word that I have not given, in every oxygenic photosynthetic organism. And that term oxygenic means they are able to use water as their primary electron donor. So that includes every plant, pine trees, algae, mosses, bryophytes, green algae, red algae, and cyanobacteria. All of those organisms evolved a very similar reaction center at least 2 billion years ago. And the molecular nuances and details from these low prokaryotes to these advanced eukaryotes is for all intents and purposes 100% the same. And we assume 100% the same quantum efficiency at that level. But the plant growth and how fast they grow, how much biomass, those are very idiosyncratic by different plants and different growth habitats. And it's also dependent upon the environment. In the tropics, plants go very efficiently because they're not so limited with things like water, so they can keep their stomates open. The stomates are open, gas exchange is very good, things like that.
Yeah, I would also like to say that if anybody wants to ask a question in Spanish, we can also do that, and then we can do translation.
Yes, we have many good translators.
Let's have a question over here. So the same, if you can do a brief introduction.
Oh, hi. Just a brief question regarding the titanium dioxide. Why is it the material of your choice?
Oh, so that's always a good question, and I cannot tell you, but we have a theory, and the theory is that there are in any of these materials there are imperfections or irregularities, and we think probably the irregularities were what were being used in the initial phage display. So the peptides were selected. We don't know what the phage display work was done by another scientist actually preceding my work. We don't know what the material was that they used to select for the peptide that bound to their titanium dioxide. So it's possibly that it was an irregularity or a lattice imperfection. We know that when we've done similar work with graphene, we started trying to make graphene binding peptides. I aborted the project because we could never push the affinity of the peptides high enough. And graphene has this edge region, and we think that the only peptides ever bound were to the edge. And the edge is not what we want. We want something that reacts with the surface, but the surface is too chemically inert to interact with the peptide. So that there are possible solutions, but it's a very, when you do phage display, it's essentially a lot, it's screening for that one unique structure, but we have no information really to tell you what in the structure it's binding to. And same with the making of the nanostructured titanium dioxide nanoparticles. My colleagues think that they just make it better. So they actually think that they make it with less defects than the commercial stuff is. So I can't tell you whether it's the lack of the, you know, the imperfections, the more uniformity. I had a grant, and this guy was a good material scientist, and so he made a lot of these things. And he was very proud of the material he made because it was so much better than anything we could ever buy.
Just merely due to like binding concerns, nothing to do with the electronic properties.
Yeah, this was a really low level screen. This was basically, does it stick or does it not stick? We didn't actually test whether it was good for conductivity and solar cell design. Right now, we're using the material that we buy from Michael Gratzel's company, Solaronics. And it works pretty well, although the stuff we make in the in-house, which is, I think, an 80-20 anatase rutile, it works just as well as the Gratzel material. But I'm happy with any material scientist to test them, because we do believe that that's, you know, that's a problem and a limitation. And as I said, and as Claudia has always kind of complained about, we are really only coating the very upper surface. So the amount of, you know, photoactive surface that we have in the just the Dr. Blade material is probably under 10% maybe. That's why we wanted to go away from these globular heat cindered materials to things that have cavities, and the cavities would allow us to actually electrophoretically drive our proteins all the way into that bicontinuous array, and that could increase our binding capacity, literally log folds. And you know, log folds, that's where we're in. You know, that was a log graph I showed you at the end, and we seem to have kind of gotten to this upper level. We're not pushing ourselves to the next level. We're up to just about 1 milliamp per square centimeter in photocurrent.
And another follow-up question. When you speak about surface and this compound, this protein, you know, like complex bound to the surface, do you envision these devices to be operating in a dry surface, or do you need also to hydrate the layer where these proteins are bound in order for them to be like in an ionic solution, or how does it work?
Yeah, so the solutions that I worked with Gratzel were in a ternary mixture. It was aqueous, acetonitrile, and ethylene carbonate. So it was an ionic it was a relatively complex media. It works. It does not work that well with PS1. It actually damages PS1 a little bit because the acetonitrile is fairly high. I have a student, Alex Theodore, who went through a very careful systematic study of solvent systems that are compatible with PS1. And so we did find a system that would work very well. But the cobalt complexes that Gratzel made and that we were making were not water soluble. So then we went to water soluble systems, and they work better and they're easier and the complexes are more stable. Now, in the first device I made in MIT, it was a completely solid state anhydrous device. So those systems, we basically put the reaction center on under ultra high vacuum. Okay, so it was essentially basically at 10 to the -6 atmospheres overnight, and so it was completely anhydrous and it worked. That system then was anhydrous, and then we sputter coated the organic electron transport layers on top of it, and so there was essentially no water in that system. I mean, an infinitesimal amount of water was remaining, and it worked. So there is no reason, if we could get the electron transport layer to be sputter coated on top of it, we could probably, if we got it in close contact, get away from the aqueous mediators or the non-aqueous mediators. And that's kind of the goal we're doing. We'd like to get the ferredoxin to bind very tightly. And now we've moved that electron out of this more nonpolar region into a more polar region. And now if we could come back and sputter code a ETL on top of that, the devices would probably not only work better, but we would assume that their longevity would be more. So everything that's bad in biology all ends with ACE. So hydrolases, nucleases, whatever, they all use water as their nucleophilic attack. And so removing water should increase the stability and longevity. So that's the goal. Yeah.
Okay, thank you so much.
Thank you. So now we're gonna go into the question from Sophia in Zoom. Yeah.
Hi, well, my name is Sophia. I am a student in materials engineering. And I was wondering, you said that like you have created one that lasted like 100 days and you were aiming to longevity as you like just said, but what happens when it stops working? Do you have to replace all of it or just the protein?
Okay, good.
Or,
It's
like,
good questions.
you-
Okay, Sophie, it's a very good question, but unfortunately I spoke a little incorrectly. And the reason I spoke incorrectly is we tested it for 280 days. Okay, you know, science goes on. And in 280 days, it was still 100% active. So we could not estimate a half-life because after 280 days, it was as good as it was on day one.
Okay, it's not like it stopped working
No, it
and then
didn't
you
stop
stopped the
working. That's
project.
I've been trying to get a student, you know, most of my students don't have long enough tenure in my lab to maybe do a careful study. They'd have to maybe do it for 5 or 6 years. I'm going to guess these samples there's two caveats to that. One is light intensity. If you give them, you know, 3 times the sun, they'll probably die faster. Most of the work we did is always around 1/5 solar intensity. And the other aspect is spectral properties, the color of the light. And I showed you that plants are green because they don't absorb between the red and the blue. The blue is the second excited singlet state. And that, if you use blue light or ultraviolet light, we know that the sample is damaged faster because the electron in that second excited singlet state can undergo essentially a triplet state, and the triplet state is biologically reactive. So if you stay with light from 500 nanometers up and stay below, say, 1000 microeinsteins, I think the devices might last indefinitely.
So you test it in the lab, you don't test it with solar, like natural light?
Yeah, in a robust way, the answer is no, although we have set these things out in the sun for a while just to mess with them. So we haven't really done these things we're not NREL and we don't have all of these facilities to set these things up. We don't even have a solar simulator in my lab. So these things are usually done with light sources that have controllable output and spectral properties.
Yeah,
So
like
it's not
control
real world yet.
the results.
Yeah,
Okay,
it's not as real as people would like.
Okay.
thank you.
Thank you, Sophia. So we're now gonna open the microphone for Thomas Sullivan.
Hi, first I wanted to congratulate you on a great presentation. Unfortunately, my own knowledge on protein and photosynthesis is quite limited. So I would like to ask a technical question, but I don't think I can.
You can try.
I'm going to try, yes. Going back to ferredoxin, I was very interested in the how you try to synthesize ferredoxin. So I've got 2 questions. And the first question is, in the synthetic ferredoxin, you mentioned there were some problems. And have you managed to find the root of those problems in the structure of the ferredoxin? And my second question is that, can you artificially synthesize natural ferredoxin, or do you have to extract it from natural means?
Okay, I'll answer your second question first. The second question is that we use a recombinant form of ferredoxin. So we actually make it in E. coli. When we make the E. coli expressing this, it makes it pink. People always say it looks like pink grapefruit juice. It's quite red, but that's because the protein is being expressed and it's putting the iron sulfur center in. We do a little bit of tricks to try to increase that process by adding extra sulfur and extra iron into the media. But we make it recombinantly and we can change the sequence whichever way we want. So that's not a problem, and the yields are quite robust. The first part of the question is a little bit more complicated. The question was, what are we going to do from the protein design perspective to try to modulate the midpoint potential of the iron sulfur cluster? And that's work that is actually at a almost at our theoretical limit. So what we have decided is that there's ligands that bind to the iron sulfur center, and we go out to the next layer. You can actually kind of think of this as like a peel. And if you go out to the second boundary layer of the coordination groups, the theory is that if you change the polarity of those, the polarity of that will change some of the electron negativity, and it will shift the midpoint potential in a predictable way. Okay? That's the theory. So we've made all of those mutations that should be shifting the potential down. And in most cases, one case they worked, and I think 3 cases it did not work. And so that just says that there are factors that modulate the midpoint potentials of metallocenters that are coordinated by protein components that is outside of our theoretical understanding. So it's disappointing. I mean, David Baker's lab is literally the most innovative lab. They are not electrochemists, so we do all the electrochemistry in our lab. But we are frustrated that we have not been able to make a complex with the midpoint potentials approaching the natural ferredoxin. Now, one other caveat with that, I will just tell you, when you start dealing with midpoint potentials in the minus 400 to 600 millivolts, they become extremely oxygen sensitive. And so if oxygen is ever in the sample, so we have to do all this work under anaerobic conditions. So my lab has to have a glove box. We actually could never ship them from Seattle to my lab in an oxygen-free way. So then we take the apoprotein into my lab and we reconstitute it in a glove box under completely anoxygenic conditions. And that has solved the problem a little bit, but we still have more problems to solve. Is that a helpful answer?
It was, thank you. And thank you for being able to interpret my question correctly. I think, yeah, a lot to think about. Thank you very much.
Okay, so now we are gonna have the question from Thomas Alejandro.
Thomas.
Okay. Can you hear me?
Yes.
All right. When you were exposing about the 3 models, about the black energies, green energies, and the artificial model, I'd just like to know for how long could be the transition from the black energies to green energies and then to the artificial model.
So what are the challenges going from black to green?
Yes, besides of besides of how long it could spend.
So I think I understand your question. I mean, one of the challenges in all of the biomass, say I live in Tennessee, and Tennessee's had a lot of money from the federal government to try to build cellulosic bioethanol. And the idea is that if you can grow a plant like switchgrass or some other cellulose-rich material, that you can essentially convert the cellulose to ethanol through microbial conversion with enzymes like cellulase, and then aerobic fermentation and things like that. The problem with all of those biomass processes is the amount of land you have to use for starters. Most of the ethanol being made in the world is in Brazil, and they're using sugarcane. And to make the sugarcane, they're cutting down the rainforest. Okay, so there's always this consequence of trying to convert land use to making fuels, and there's never really going to be a very good answer to that. The second thing is that a lot of the inputs people are not paying attention. Biomass production is always very intensive in water use. So the water use, which as you can see in the United States, we have half the country in a 500-year-old drought. This is the biggest drought in 500 years. Some people say maybe in 1,000. So we're not going to have the water to be allocated to growing biofuels crops. So I think that's one of the biggest problems. You know, historically, people in the US have been taking corn, and they've been taking the starch in corn. This is corn that probably we as humans wouldn't eat, but animals would eat it. And so you're still diverting food to fuel, and that's considered to be a very unacceptable transition as well. So I'm a very anti-Black person. When I say anti-Black, I mean anti-fossil fuels. But I used to have a slide that we shifted the Black to brown, which was to be biomass, and I'm pretty much an anti-brown biomass person as well, because I think all of those systems are unsustainable, and most of them are not even economically viable. Now, our technology is neither sustainable yet, and it may never be, but at least it's an idea that we think we can push it forward. I feel there's a lot of people that ultimately feel the solar energy technology that is ultimately used is going to have either direct parts of nature, or it's going to be directly designed around nature. So the biomimetic people want to go in and find out what is the redox centers that are able to split water and build a catalyst that's around that same enzyme design. And that's very challenging. It's been unfortunately funded a lot more. The biomimetic people, or the artificial people, have about 100 times more money than the applied photosynthesis people. So it's disturbing. Their challenge, they can make a catalyst that works 50,000 times. Their turnover numbers can be 50,000 times, and they can work for 4 to 5 minutes. Whereas we make systems that have turnover times in the millions and works on the time for months. So we just don't have the sex appeal for some reason. Okay, did that help? Did that help?
It's okay. Thank you very much.
Sure.
Thank you, Thomas. Okay, now we have a question from Sebastian.
Hello, first of all, thank you very much for this very interesting presentation. My name is Sebastian. I'm a student in material science and engineering. My question is regarding the study parameters that you used in your cells. Regarding, you said you used 1/5 of solar intensity, is that right?
Yeah, I mean, typically our longevity studies were with 300 micro-Einsteins of light. So where we live, that's about 1/5. But for you guys, maybe it's a little more like 1/6. You know, you're closer to the equator. 300 micro-Einsteins.
Sorry for the noise, for the background noise. So yeah, my question was regarding why was this parameter decided, but now you answered it. It's because of your location.
No. It's really hard to get light sources that have the intensity of the sun. Okay, I mean, that's the practical reason, is that we want something that works. So we have an LED, and an LED can basically, we run it at full intensity, and we have these fiber optics that focus in, and we actually have a photo detector underneath our system, so we can measure it. We just didn't have the light to go much above that. I think we could max out at about 700, but we ran this at 300.
Oh, okay. So it's related with the limitations of the devices that you had?
Yeah, I mean, it's probably twofold: limits by the device, but also we wanted to run this. We wanted to have a sample that would work for as long as possible, and we already had data that would tell us that if we give it really bright light, it damages it, and if we give it blue and ultraviolet light, it would damage it. So this was essentially 500 nanometer and up, sort of green to red light. So we already had that. Now, that's not a real problem. I mean, people already are designing solar cells where they put in essentially a optical filter to cut out certain lights, just like some cases they do it combined with anti-reflective coats so that that doesn't reflect the light. So there are existing solar cell device designs that can give us spectral filtering. We also, as maybe this isn't something I talked about, is that photosynthesis, even though the process is very similar, nature has developed photosynthetic proteins that harvest light from 450 nanometers all the way out to 1100 nanometers. So we think we could use different biological components and essentially build a sort of a multi-junction device that each layer filters out the useful light and transmits the unuseful light to a second device and a third device and a fourth device.
Okay,
So,
thank you very much.
Yeah.
Yeah, thank you.
Okay, so we're gonna have one more question because we need to close the session. We're gonna open the microphone for Professor Enrique Quiroga.
Hey there, good afternoon. This is Enrique from the University of Puebla in Mexico. It was a very nice talk today. Very encouraging, I will say. It was very nice. I enjoyed it. I just have a question here. You presented a couple of examples where you used as support, for example, zinc oxide or titanium dioxide. And these materials, we know that they have a large band gap. No, they absorb in the full range, I would say, or they are transparent, I would say. No? I wanted to ask you if there is another characteristic besides the band gap to avoid the absorption, to use it as an inorganic material for this application, not just zinc oxide or titanium dioxide, if you have thought about other materials.
Yeah, I cannot tell you everything about that, but I know that there are a lot of the metal oxides, lead oxide and tin oxide. There's a lot of people that are very interested in going to these other materials because of some of their properties, the size of the band gap, the sustainability of the natural ingredients, and some of the fabrication tools. Not being a material scientist, I'm not really confident that I would be able to tell you the best material. We went with titanium dioxide because the foundational work that's already been done in dye-sensitized solar cells is so good. There are many, many tricks and protocols on how to make really good mesoporous, heat-sintered titanium dioxide. The growing of the nanotubes is an art. We've tried in my lab, not with much success. We can grow them, but they don't have any regularity. So you have to use other more sophisticated epitaxial growth techniques and things like that, which I'm very robust to use. It's just as a molecular biologist, I can't make those devices myself. If we have somebody like yourself that can help, I'm very happy to work with other material scientists.
Yeah, it is great. Yeah, because I was wondering about the properties, for example, from the properties of the surface of the material. If you need certain groups, there are functional groups to be able to use the material, not just the properties of the material in the bulk state, like the band gap, but maybe other properties that you may need for this application.
Yeah, there's a lot of, there are chemistries for coupling materials directly to these metal oxides that are already in the literature. There's some relatively robust covalent bonding chemistries. Most of those are not using proteins though. So we have to, if we're going to do some of that, we have to have chemistry that couples the linker to the metal oxide and also the linker to the protein. That's why we went with the random phage display, because now we find a proteinaceous material that a molecular biologist can use and apply it directly to the protein. As the device materials change, just like our question earlier, the affinity of that peptide may change as well. So if we make a new material, we may have to go back and evolve or select for a new peptide motif. Peptides are beautiful because with 21 amino acids, you have nearly an infinite range of surface chemistry properties. Even with only something with 7. So if you do 21 to the 7th, that's a lot of variability available to your design. Nice. And just one final thing, we have now moved into the area in synthetic biology where we're using non-natural or non-native amino acids. So we can make a protein not with the amino acids that you and I have, but we can make it with a completely new amino acid. And these new amino acids can have chemical properties that are completely unique and almost no limitation to our creativity. We just make a new amino acid and we use this synthetic biology to basically put it into the protein wherever we want. So that's really at the front edge of synthetic biology, the incorporation of non-natural amino acids. And non-natural amino acids can have properties of whatever our interest. And a lot of them have photoactive cross-linking abilities so that if we shine light on it, it builds a covalent bond with whatever is within 2 or 3 angstroms.
Okay, thank you so much.
Yeah.
Ciao, ciao.
Okay, thank you. Okay.
Okay, well, thank you everybody for being here today, for joining. This is such an interesting experience also to do a hybrid event and interact with the people online and in person. I would like to do a final observation that this is a very complex field. We have seen how we need a lot of interdisciplinary work, all the way from physics, computational, engineering, molecular biology, and to motivate students, that we have many students connected now, and to consider within your career paths. And I think that Professor Bruce would agree with me that whatever it is your major, there is a space for you to work in this field and help the advancement, because it is very complex. What do you think, Professor?
Yes, I think it's going to take many people, and there are no shortage of challenges, but the best ideas probably haven't been even thought of yet. So anybody who wants to get into this, there's opportunities. And, you know, even if we never actually make a green solar cell that's going to do this, we'll probably make something. I will just tell you one kind of sort of disappointing. My very first grant to work on this was from DARPA, the Defense Advanced Research Projects Agency. And they didn't want to make solar cells. They wanted to make PS1 to reduce the size of their laser guidance missiles. And so we may be able to make cameras based on PS1. We can make possibly solid state switches that work on nanosecond to picosecond levels that are driven by PS1 and light. We may be able to make all sorts of sensors that are associated, because PS1 is such a remarkably fast photon detector.
Yeah, certainly a fascinating world of protein and materials hybridization. So thank you, everybody, and let's give a final applause for the professor.
Thank you.
Now before we finish, we would like to give you a certificate. Yeah. Y el papelito, lo tengo yo. Okay.